Hello students, this is Inheritance and Variation Part 7. In today's session, we are going to understand two crosses, that is what is test cross and what is back cross. So what is a test cross and back cross? To test the unknown genotype, back cross is used. Okay, to test the unknown genotype, we use back cross. Test cross can be used to find out the genotype of any plant with dominant expression, but it is not known whether it is pure or hybrid trait. That means whether that unknown genotype it is capital T small t or it is capital T capital T. Okay. So in this case, it is crossed with the recessive plant with that particular trait. Test cross can be defined as a cross between individual with unknown genotype for a particular trait with a recessive plant for that trait. So test cross is a back cross, but back cross is not necessarily a test cross. So what a test cross or back cross, what do we mean by that? So test cross is a single method. Okay, it's a simple method which was devised or given by Mendel to verify or to test the genotype of those plants which we get in the first filial generation or in the second filial generation or in the third filial generation. So all in the F1, F2 or in F3, all in all these cases we get all hybrid type of plants. So to verify their or to test or to find out their genotypes, Mendel devised a simple method, that is test cross or back cross. So when hybrid expresses recessive trait, that means in the dwarf pea plant, the genotype is definitely small t. But if the hybrid tall plant, if we consider, it is not possible to know its genotype. Okay. So that tall plant will be a capital T. It will it can carry capital T type of genotype, or the tall plant it also can contain capital T and small t. So that tall plant it is homozygous dominant or heterozygous dominant. So we cannot surely tell or express or find out the genotype. Okay, whether it is capital T small t, whether that plant is homozygous in short or heterozygous. So to test, okay, to find out whether the tall plant or any dominant expression is homozygous or heterozygous, test cross can be performed instead of selfing. Now you can observe this image. Okay, in this image, this is a monohybrid cross. In this image, this is parental generation. Okay, the tall plant, it is homozygous tall plant as it carries capital T capital T. It is crossed with dwarf plant small t small t. Again it is homozygous recessive trait. When the homozygous dominant plant is crossed with homozygous recessive plant, in the first filial generation we get all tall plants. Okay, but these tall plants are not homozygous but they are heterozygous. Okay, that is capital T and small t. But for example, we or suppose we don't know what type of genotype this tall plant carries. Okay, so whether the plants which we get in the F1 generation, these are homozygous or heterozygous, to find their genotype, that whether these tall plants they are having capital T type of genotype, or these tall plants they carry capital T and small t, that is heterozygous type of genotype. What we have to do is we have to cross this tall plant from the F1 generation with this dwarf plant, that is with the homozygous pure trait, homozygous recessive plant, and then we will understand, or then we will find out its genotype correctly. Now you will understand this example in a better way in the next slide, in the next explanation. Okay, so now moving to this part, you can observe there are two diagrams. Okay, now in the monohybrid test cross, we don't know the genotype. Okay, so homozygous tall plant, this is pure tall plant. It is crossed with homozygous dwarf, that is pure dwarf. Okay, and it forms these type of gametes. Now it forms one type of gamete, that is capital T. It forms pure dwarf plant. It forms one type of gamete, that is recessive, small t. So it contains heterozygous all tall plants. But we want to we want to verify the genotype of this heterozygous or genotype of this F1 type of plant. Okay, so what we will do in the parental generation, we will take this plant which is having which belongs to the first filial generation. Okay, so we will take this capital T small t or whatever F1 generation type of tall plant we are having. So we'll take that tall plant as in the parental generation. Okay, now this tall plant, and we don't know its genotype, its genotype is unknown. Okay, so now this tall plant is crossed with the recessive dwarf pure homozygous trait. Okay, so what we will get, it will form gametes. Okay, it will form capital T small t small t small t. Now in the F1 generation, what type of combinations we will get by plotting the gametes in the Punnett square? We will get now this capital T capital T. Okay, this is capital T capital T. This is small t small t. We are plotting the Punnett square, and this tall plant is crossed with this small t and small t. Okay, so what type of combinations we get here? Capital T small t, this is first combination. This plant is tall but it is heterozygous. Another is small t, that is one plant is dwarf. Another capital T small t, it is again tall heterozygous, and another is small t small t. So we are getting heterozygous tall plants. They are two heterozygous. Okay, these are the two heterozygous tall plants. How will know? How will find out that, or how will we suggest that these tall plants are heterozygous or not and not are homozygous? Because we are getting homozygous dwarf plants too. Okay, so we are getting homozygous dwarf plant. That means the small t is also getting expressed with the capital T. Okay, so we are having here two tall heterozygous plants with two dwarf heterozygous plant. That is, their ratio is 1:1. So in the dominants and recessives, they are 1:1 in ratio, and that ratio is known as test cross ratio. That is 1:1. So what we exactly did in the test cross? In the test cross, the F1 hybrids for or F1 is crossed. Now this is F1. Okay, F1 tall plant is crossed with the recessive homozygous recessive parent. Okay, that is small t parent. Since the offspring is crossed back, now we are crossing back this F1 generation with this homozygous plant. Okay, so it is back crossed with one of the parents. It is called as back cross. So to test something, we are crossing back. So test cross is a back cross. Okay, so what we did exactly in the monohybrid test cross, in a monohybrid cross of pure tall and pure dwarf plant, all F1 hybrids are tall. Okay, but with the genotype capital T and small t. To verify their genotype, when they are crossed with the recessive parent of that progeny, will be 50% tall and 50% tall. Now here we are getting 50% tall plant and 50% dwarf plant. Okay, this is because recessive parent will produce only one type of gamete, that is small t. Okay, it will form only one type of gamete, that is small t, and the hybrid will produce two types of gametes, that is capital T and small t. Therefore half of the progeny will have genotype capital T small t. Now capital T small t and half of the progeny is carrying small t and small t type of genotype. Thus dominants and recessives are in the 1:1 ratio. Okay, this is called as test cross ratio. So the cross between F1 hybrids and the homozygous tall parent is also a back cross. if we suppose we backcross the this F1 progeny tall plant with this pure tall, that is pure homozygous tall plant, which is carrying a genotype capital T and capital T. Okay, so if we cross such back cross, now as we are crossing back it again with the dominant trait, not with the recessive trait but with the dominant trait. So as we are crossing it back, so it's a kind of back cross, but this will produce all tall plants. Okay, as this is a pure dominant trait and this is heterozygous pure dominant, it is heterozygous dominant trait. Okay, so we will get 50% pure tall and 50% hybrid tall. Therefore, backcross with the dominant parent cannot be used to test the genotype. We will get all the pure tall plants, but we cannot get the genotype. Okay, we cannot test the genotype of this F1 plant or the hybrid plant. So test cross is a back cross where we cross back. Okay, so test cross is a back cross, but back cross is not a test cross. Okay, so let's move to the next. Now, why the back cross is important? The idea of backcross, the idea of backcross is to improve the breed of an organism. Okay, so we will take couple of examples. Suppose we have one organism which is heterozygous dominant. Now this plant is carrying capital T and small t, so it is heterozygous dominant, and another plant that is homozygous recessive, which carries the genotype small t and small t, and we want that in the next generation, that is in the F1 generation, the recessive trait should not be expressed, or it should get eliminated. Okay, so that is our motto, that is our objective or aim. So to improve the homozygous recessive trait, this trait homozygous recessive, what we do is we will cross this to improve this homozygous recessive, we will cross this homozygous recessive trait with this homozygous dominant. Okay, so homozygous dominant trait is crossed with, or this genotype when it is crossed with homozygous recessive, that is small t type of genotype. Now what is our agenda, what is our motto? That we don't want this capital T, we don't want this small t, we want to eliminate this homozygous recessive trait. Okay, because we want to improve the breed, we want all the tall plants, whether the tall plants are heterozygous or homozygous, but we want to eliminate this small t type of genotype. So what we will do is we will cross that recessive homozygous trait with the homozygous dominant trait. So by plotting the homozygous or heterozygous traits in a Punnett square, what we get is capital T, okay, and small t. What type of genotype we are getting here? Capital T, small t. Okay, now this is capital T, small t. This is capital T, small t. Again, capital T, small t, and capital T, small t. So all the four progeny, whatever the plants we are getting here, all are heterozygous dominant traits. Okay, so the recessive trait is not expressed, or it is disappeared. Okay, so small t type of homozygous recessive character is completely disappeared. Okay, so that is our agenda. So this homozygous dwarf will be crossed with the homozygous tall trait. So we will plot a Punnett square. Okay, and in this Punnett square, what we get? That all the traits are tall. Okay, and small t type of genotype is completely eliminated, or it is not expressed in the F1 generation. Okay, so backcross is to improve the breed of an organism. It is also used for that mechanism. Okay, so another example which is given in your book is to understand the backcross and test cross, or to understand the genotype, how we come to know the genotype, how we find out the genotype. For example, see, now this is a dominant phenotype. As you can see, the genotype is unknown. Okay, Here we are having a violet color flower. Now this violet color flower is a dominant trait in the Pisum sativum plant. So there are two chances that this violet color flower, it will carry capital R type of genotype or capital R, small r type of genotype. It will be homozygous dominant or heterozygous dominant. But as the genotype is unknown, we want to verify it. So what we do, we will back cross it. Okay, we will back cross this capital R with this small r, which is a homozygous recessive trait. Okay, so when we back cross this capital R, that is homozygous dominant trait with this homozygous recessive trait, we get such type of result where capital R small r, that is capital R small r, capital R small r, again second violet color flower, then capital R small r, third violet color flower, and capital R small r, that is all flowers are violet. So unknown flower is homozygous dominant. In case of this type of cross, when we back cross the violet flower with the homozygous recessive flower, and if we get in the F1 generation, if we get all violet colored flower, that results in the homozygous dominant. That means this flower is carrying homozygous dominant type of genotype. Okay. Okay. In the next case, if we cross this capital, if the genotype of this plant flower is capital R small r, that means if the violet flower is not homozygous but it is heterozygous, okay, dominant, then if we test cross this type of phenotype, that capital R small r is when crossed with the homozygous recessive trait, okay, that is with the white flower which is carrying a genotype small r small r. So what type of result we will get? Capital R small r is its genotype as it is heterozygous, okay, as we don't know its genotype. If it will carry capital R small r type of genotype, when we will cross it with the small r, that is homozygous homozygous white colored recessive trait, then we will get two violet color flower and two white color flowers. Okay, as small r is present in the heterozygous trait, okay, in the heterozygous dominant violet color flower. So this small r can get fused with this, okay, it can get expressed with the small r, okay. So small r will form small r genotype homozygous, that is one white colored flower, and small r with another white colored flower. So half of the flowers are violet and half of the flowers are white, okay. So if that genotype of that flower is unknown, and if we want to find out, or if we want to verify the genotype of the violet color flower which is phenotypically violet, but we don't know whether it is homozygous dominant or heterozygous dominant, what we will do, we will test cross it with the recessive homozygous recessive type of trait. If that flower will be homozygous dominant, then all flowers will be violet in color, okay. So if in the by hybridizing, by crossing of these two plants, if we get all plants with violet color flower, that means it is having homozygous dominant genotype, okay, capital R, okay. And if the by crossing the same plant with the violet colored flower, if we get half of the progeny with violet colored flower and half of the flowers are white in color, that means that type of plant is carrying capital R small r type of genotype. That means it is a heterozygous genotype which is present inside this violet color flower, okay. So that is a graphical representation of a test cross, where we test cross the dominant phenotype, okay, with by crossing it with the homozygous recessive trait. So that's all for today. In this today's session, we there are two concepts, okay, that is one is back cross and test cross. So if the question is asked on the question can be asked on separately on back cross and test cross, or a question can be asked like all the traits or all the test cross is a back cross, but why back cross is not a test cross? Okay, so such type of question can be asked. So if uh, these questions are asked for 2 marks or 3 marks, you have to draw the diagram, you have to draw the Punnett square, you have to write in detail about the phenotypic characteristics, okay, parental generations, what is homozygous trait, what is heterozygous trait, okay, and again the F1 hybrids, right? And of course you have to draw the Punnett square, the gametes and their combinations. You have to write the uh, genotypic ratio, okay. In the monohybrid test cross, you have the genotypic ratio is 1:1, okay. That is a test cross ratio that you have to write in the answer. So that's all for today's session. Thank you.